So I'm going to talk to you about better science for future us. And I'm a scientist who has worked for a rather long time at the interface between the biological and computational sciences. And I wanted to share a few vignettes with you about my personal path to open science in a way to help you see and imagine how making our science more open in small steps with kindness can have a huge impact on future you and future us. And through this path, I've come to feel like the data world is my oyster. I've had some what I actually call pretty euphoric collaborations with colleagues and friends. And one of my favorite concepts in evolution is that of contingency, meaning something that could happen as a result of something else. And so I've put a couple of rocket ship emojis in my slides where I want to point out people and things that influence my path and consider you can recognize those people and things that are in your paths as well. And for better or for worse, a lot of our success can be measured in or has been measured in publications. And so in a few places, I've added publications of mine that have come as a direct result of my engagement with open science. So immediately after my master's graduation, I was working as research assistant in a lab. And my task was to clone and sequence, this is pre-full genomes, the N-terminal 500 base pairs of a pine tree gene. And through, luck or contingency, there was this visionary grad student who had installed web browsers on all of our lab computers. And this was back in 1994. And what this gave me was access to the public GenBank database, where I actually found sequences of this gene from many other plants. And this launched me into a whole new research question and a realization that I felt I could learn more about my favorite enzyme and faster by comparing sequences from public databases than spending the time sequencing yet another little 500 base pairs. And we managed to upend some dogma in the process. And this led directly to my first author publication. This is the interlude where I give a shout out to the value of good leadership. And once I had that initial experience with public databases, that was it. I was hooked. I moved from the bench to the laptop permanently. And I was lucky to work with a man named Francis Willett, who has been instrumental in establishing bioinformatics education and research momentum in Canada, which has in turn shaped education models. And this is credit to Francis, who has this sort of dogged embracing and promotion of open science and has a really big influence on my career. So when I was working with him in this bioinformatics core facility, there was a Huntington disease researcher who came down the hall and said, hey, Steph, you guys are supposed to be smart. Can you find all the human genes that have CAG repeats encoding polyglutamine tracts? And this is relevant to other neurodegenerative disorders. And so as contingency would have it, I was a week later starting to take a Cold Spring Harbor course called Bioinformatics, Writing Software for Genome Research. And it turned out that this had the answers to the question. On the very last afternoon of the 2-week workshop, you and Bernie introduced this new Ensemble Perl API to access human genome sequence annotations in a MySQL database. And this absolutely blew my mind. It was the answer that I needed to solve this problem. And this is where I first learned about version control. But in this case, it was for genomes, where we normally think about this as code on GitHub, but this was version control for genomes. And again, this led directly to another research paper. So in my final act of working as a research scientist, for one paper, we curated post-translational modification data from 4 different methods in 15 papers. Most of that had been hidden in supplemental spreadsheets and PDFs, common theme. This was a lot of work, and many people don't necessarily have the knowledge and resources to do this. And why should they have to if we already did the work and it's been paid for by public funding? So we said, why not make this freely accessible in a single spreadsheet that any other researcher can use? And my famous last words were, this will never get published in PLOS Computational Biology. It's too simple. And then it got published there. So through all of this and just despite great support in some labs and not necessarily others, I still often felt like I was on my own. I wanted some company in these kinds of feelings, and I wanted other people to be able to share this joy too. And so gradually it just showed up as me like being interested in building small open communities in response to specific needs. And my focus really evolved from being about me to being about us. So in 2002, with Francis Willett and Ryan Brinkman, we founded the Vancouver Bioinformatics User Group, or VanBug. Is a citywide seminar and networking series where we had audiences of up to 150 people. And this is still going on after 20 years. People made friends, people got jobs, they found PhD supervisors, collaborators. And in fact, one of our February 2013 speakers was Gavin Ha. So tell him I said hi. In 2016, I made the move to like officially community building as a profession in science. I was our Open Sci's first community manager, and I can tell you lots about that organization anytime you ask, or in Call 4, where we talk about open communities. And this is where I first used GitHub for discussing open ideas and soliciting community input on open ideas about projects to do and how to do things. But in about 2019, I started to feel like a bit of a, not exactly an imposter, but maybe a bit of a poser, and I needed to learn more R. But I didn't want to do it alone. I live in a small city. I'm in Kamloops, British Columbia. So I formed a local co-learning group. And really, this was like 4 people usually, 6 if we were lucky. But this is where I made my first XY plot in R and my first R Markdown report. Talk about sharing imperfect work. I've put a link to it there, and I'm actually proud of it. I was also invited to give talks at Fred Hutch and Our Lady Seattle on how I learned how to do this stuff, or how we self-taught in a co-learning group. And now I feel like all of this is really coming together as I get to work with the Openscapes team and sort of bringing some of these things to other audiences.